Hello, everyone. My name is Derrick Meyer, and today I am going to talk to you about CRISPR and gene editing in human embryos. So, you're asking me, what is CRISPR? CRISPR is a gene editing technology that has created a lot of excitement. But along with that excitement came a lot of ethical debate. I'll be focusing on how CRISPR works, whether it should be used to prevent genetic diseases, and whether it should be allowed to select or enhance traits in human embryos. Now, you're asking me, well, that doesn't tell me very much. CRISPR is also a tool that scientists use to edit DNA, which is the genetic code that determines how living things grow and function. The system originally came from bacteria, which use CRISPR to defend themselves against viruses. Now let's look at the biological background. How does CRISPR work? In the lab, CRISPR works like a pair of molecular scissors. Scientists design a guide molecule that finds a specific DNA sequence, and then an enzyme will cut the DNA at that spot, acting like a pair of molecular scissors. After the cut, the cell repairs the DNA, and scientists can use that repair process to remove, replace, or add genes. When CRISPR is used on human embryos, the changes affect every cell in the future person's body, and those changes can even be passed on to future generations. That's why this topic raises so many ethical questions. The ethical question I chose to focus on today was preventing disease versus enhancing traits. So let's start with preventing genetic diseases. Conditions like cystic fibrosis, Tay-Sachs disease, or sickle cell anemia are caused by specific gene mutations. In theory, CRISPR could fix those mutations before a baby is even born. Many people see this as a positive use of the technology because it could reduce suffering and improve quality of life for future generations. But the debate becomes more complicated when CRISPR is used not to prevent disease, but to select or enhance traits. Things like height, eye color, intelligence, or athletic ability can all be affected and be kind of chosen by the parents through the CRISPR technology. And when this happens, this is sometimes called designer babies. Even though we don't fully understand how complex traits work, the idea raises concern about fairness, inequality, sorry, I got a phone call, and whether parents should have that level of control over their children's genetics. This creates an ethical dilemma because the same technology that could cure these diseases could also be used in ways that change society in unpredictable or unfair ways. Now, how am I going to analyze this ethical question? I chose to go over the benefits and the risk. So let's start with the benefits. The benefits could eliminate those certain genetic diseases that I talked about, like the Tay-Sachs disease and sickle cell disease. It could also reduce long-term healthcare costs. Could improve quality of life for future generations. Think about it. If we have less of these diseases and less of these genes that cause those diseases running around in our society, it's going to be so much better for everyone involved. We could also eliminate non-desirable traits like people with red hair. Now let's talk about the risk. Edited embryos could have unintended genetic consequences. This is such a newer product, a newer lab exercise, I should say. We do not know the full side effects of this, and we probably will not know for a couple generations down. So I think that's a big risk. Mistakes could be passed down to future generations. If the machine makes a mistake, if the surgeon makes a mistake, whatever, type something in wrong, whatever, that mistake is going to be passed down to future generations. It could increase social inequality if only the wealthy families can afford enhancements. They can affect their children's intelligence, making the wealthy family's kids more intelligent than the not so wealthy family kids. So they're going to get that higher up starting straight out of the womb. And it could also pressure parents to optimize their children. Another ethical perspective focuses on individual rights. A future child cannot consent to having their genes edited, especially for non-medical reasons. Some argue that choosing traits for a child takes away their autonomy. There's also the societal impact. If enhancements become common, society might start valuing certain traits over others, which could lead to discrimination. And nobody likes discrimination. So, my position on CRISPR gene editing in human embryos: CRISPR should be allowed for preventing serious genetic diseases, but not for selecting or enhancing traits. Preventing disease has a clear medical purpose and can reduce suffering. However, using CRISPR for enhancements crosses into an area where the risk, social, ethical, and biological risk, are way too high. We don't fully understand how complex traits work, and trying to engineer them could create more problems than it solves. I also think that allowing enhancements could lead to unfair advantages and widen the gap between people who can afford genetic technology and those who cannot. So, in conclusion, CRISPR is a powerful technology with the potential to change medicine and human biology. It works by editing DNA at precise locations, which could help prevent genetic diseases before a child is even born. But when it comes to enhancing traits, the ethical concerns become much more serious. As a society, we need to carefully consider how far we want to go with genetic engineering and what kind of future we want to create. Here is some of my sources, and thank you guys for listening.